Hey AP students, today in our video we're actually going to be talking about lab techniques for separation of solutions, and it also goes right along hand in hand with solubility. So let's go ahead and kind of just jump right in. So our first one is filtration. Filtration is obviously we're separating a solid. All right, wow, that is very large. Sorry guys. All right. Filtration is we are separating a solid from a liquid. All right. A lot of times when we do this, we will use a funnel and filter paper to do this. And our filter paper would be right there, and it would separate it out, and we fold our filter paper. It's actually very kind of cool. Our filter paper starts as a circle, so then we fold it in half, right? And then we fold it again in half, so we've got our fold, and then we fold again, and so now we've got kind of like this little thing that looks like that, and then we just open one end and we put it inside the filter, and that's how we filter. You can also, most of this works via gravity, right? We're talking about gravity, letting just kind of gravity pull it down. You can also have suction ones where you set it up to a suction and it will help it go through faster, because a lot of times it takes a little bit. The other thing that you want to do with this is, since we're separating a solid from a liquid, our key here is we need the liquid to dry, right? The liquid will need to be dry before we can mask that filter paper. So a lot of times you'll have to dry it overnight or maybe over a couple of days to get the correct amount, or the correct, like if we're trying to figure out how much solid we have. Okay, so that's the first one. Another way to separate our solution or a mixture is via distillation. And distillation is basically removing impurities from a liquid. And this is by boiling. We're doing this by boiling it. So what we have is we have kind of a setup where we have two flasks, and on one side we have it heated up, and we heat that up to the point of evaporation. And when it evaporates, it's only the pure substance that evaporates. And so then we have a condensing tube that actually cools it down, and then it gets back to liquid again. So the stuff in the second flask, in the cooled flask, is pure, and then our impurities stay in that initial flask. The one we're going to have experience with in class and actually do together is chromatography, and chromatography is our separation of colors. All right, so we're going to be separating by colors, and this works, chromatography works, because like dissolves like. You've now heard that term a couple of different times, but like dissolves like means polar substances dissolve polar substances, right? So thinking back to like when you get a grease stain on your shirt, right? And you throw it in the washer and you throw it in with your detergent and you're like, oh, see you later. But yet the grease stain is still there, right? When you pull it out. But if you use a little dish soap on it, boom, it'll be taken care of and you have it. And that's because like dissolves like. Detergent most of the time is polar, right? And when we, our grease stain would be non-polar, and so they don't dissolve each other. And water we definitely know is polar, so that doesn't work for us. All right, so when we do this, what we have in chromatography, our basic gist of chromatography is we have a way to separate them out, and usually it's via gravity. All right, and we have like a polar solvent, right? That means that's something we're setting it in. And so let's just kind of take an example of paper chromatography right here. So I take my little, here's my beaker, right? And I put a little bit of my polar solvent in the bottom, and then I will have taken a piece of filter paper, and I just drop this in here, and then via capillary movement, it'll go up the paper, and we'll have separation that way, because like dissolves like, as well as the difference in molar masses. But again, it's a little bit, it's more about like dissolves like. So if our filter paper is non-polar, right? So our polar stuff is going to kind of stay down at the bottom and move slower up the line there. So paper chromatography, that's what we've got. We've got separation on filter paper, right? Thin layer chromatography is very similar to that, except you're using like silica gel or something like a medium right there that gives it a chance to move through there. It looks like filter paper, but it's not quite filter paper. And you can actually do this one with like substances that are that glow under a UV light. That's where I've had experience with it. And then column chromatography, you have like a big old solution and you can do this and separate it out. And you can see it moving down the column in a band. And so you have a slurry inside there of usually silica gel, and it separates it out like that, because based on what you're pouring through and what the slurry is made of, it'll determine your polarity. And if you do it, it'll separate out, provided the polarities are just different enough. Okay, so now going to our data, when I do this is paper chromatography, but this would work for all of them. So here's my filter paper at the end, right? I have my solutions separated out. We can see the red moved the most, then the green, the blue, the yellow didn't move very much. Our solvent level was right there. We started them all on the same line. That's our base height. And what we do to figure out like our RF so we can compare them, we compare them by taking like the middle, that's the middle of our dot there, and we take that distance over how far the solvent front moves. So it's a ratio. So if I did it and I just maybe didn't have time to completely finish it and let it run, our ratio would still be the same because the solvent front would not have moved as much. All right. Again, hopefully we get more experience in this when we do this in class, but that's a kind of a quick overview of how we would collect data. All right. See you next time.